Hello and greetings to you, Prof. My name is Nadatul Aisyah binti Abdul Aziz. Today, I'm going to present to you the genome sequencing project research article that I have chosen. Nontuberculous Mycobacterium (NTM) are mycobacteria other than Mycobacterium tuberculosis and Mycobacterium leprae. It can also be referred to as environmental mycobacteria. It is an ubiquitous microorganism which can be found in our surrounding. Nontuberculous mycobacterial pulmonary diseases, which caused by the NTM, is increasing in developed countries. NTM typically not transmitted from person to person. However, person-to-person transmission of Mycobacterium abscessus has been reported in patients with cystic fibrosis diseases. Because of this report, it is raising concern due to the fact that NTM might be transmitted not only among immunosuppressed individuals but also among immunocompetent ones. So the objective of the genome sequencing project in this research article is to examine transmission patterns in 3 pairs of cohabiting patients with NTM-PD who were suspected of patient-to-patient transmission by investigating NTM genome through NGS, next generation sequencing. The methodology first is started with the patient enrollment. The patient enrollment criteria is as follows in the slides. Sample collection and NTM isolation. NTM are isolated from participant sputum or bronchial washing specimen. The environmental specimen was also collected. DNA preparation and sequencing. DNA was extracted from the supernatant using the Kia mDNA mini kit. DNA then was sequenced using the Illumina height sequence or the Nova sequence instrument. The last methodology is the variant calling and phylogenetic analysis. Kraken-2, a taxonomic classification system, is used to identify reads from each isolate mapping to the reference genomes, and then isolate assigned by the Kraken-2 as non-NTM species were excluded from further analysis. Variants were called using SAM tools and BCF tools. Variant calling and phylogenetic analysis. The phylogenetic analysis was conducted using the MegaX application. The results. The patient characteristic. The table shows the patient characteristic in terms of the sources, the NTM species, habitat, and radiologic findings. This table shows the characteristic on the environmental specimen. And based on the KMER-based taxonomic classification of NGS, isolate from patient A and B were identified as Mycobacterium avian subspecies hominesis. Isolate from patient C, D, and E were identified as Mycobacterium intracellulae. One isolate from patient F was identified as Mycobacterium abscessus. Based on the distribution of SNP allele fraction, all isolate were monoclonal except for the isolate from patient C, which consists of 2 clones at an 8:2 ratio. This graph shows the pairwise SNP distance between the isolates of the patients. Phylogenetic analysis was performed using the high confidence SNP from the 81 Mycobacterium avian subspecies hominesis genome. So it showed that isolate from patient A and its replicate clustered with the specimen from the kitchen, isolate from patient B clustered with the isolate from the bathroom and the kitchen. It can be shown in this. The blue one highlighted on the below one show that patient A clustered with the kitchen, while patient B clustered with the kitchen and the bathroom. This dendrogram show the phylogenetic analysis with the 17 Mycobacterium intracellular genome. It showed that all 3 isolate from patient C, D, and E were not closely related to each other. So the discussion. Among all the 3 pairs, no patient pair had near identical NTM isolate, which show therefore that patient-to-patient transmission is less likely in the study. Household water sources are the main major reservoir for NTM. It was shown that patient A seemed to acquire NTM from the kitchen while patient B acquired NTM from the bathroom. Patient living in home 2, patient C and D are farmer, so it is presumed that they acquire NTM from the soil. And from home 2, the sequencing width of NTM isolate from patient E and F aligned to NTM reference genome, but those from environmental specimen isolate did not. So it is presumed that these environmental isolate were not closely related to NTM in home 3. The results suggest that patients with Mycobacterium intracellular pulmonary diseases, patient E in home 3, may have been exposed to NTM outside the house. Conclusion. NTM isolate derived from the 3 pair of cohabiting NTM-PD patients were determined to be different strains based on the whole genome sequencing data. The sources of Mycobacterium avium in patients in one pair was identified. One acquired infection from the kitchen and the other from the bathroom or the kitchen. The likelihood of patient-to-patient transmission appeared minimal in these 3 pair of NTM-PD patients. Thank you.